Okay, let's go ahead. We'll get started.
All right. So it's a real pleasure to be here today. It's a good escape to the rain, if nothing else. For you guys, that is. I'm having fun, of course, telling you about some of the ability we've been up to in the lab. But I'll start with this quote that I think applies to all microscopy that I hold very dear from J. Baker, the naturalist. That the hardest thing of all to see is what is really there. And the way we interpret this in my lab is twofold. One, be very careful with our imaging. Be as certain as we can of the physics that are happening in the microscope and understand the transfer function from sample to whatever data we're looking at. The second part is we don't rely on human senses as much as possible. We use algorithms to look at large amounts of data in robust ways. But at the same time, we're not trying to do cookie cutter analysis. We customize or build novel algorithms in a problem specific way. And so that's our interpretation of this idea. A little bit more background about who I am and what we do in my lab. Our main goal is understanding this thing, right? The heart, it pumps beautifully for a 100-year lifespan, coordinating 3 billion muscle cells. And I'll tell you a little bit more about it, in fact, a lot more about it in the next few minutes. But to understand that we operate more on these scales of where are the molecules of bioactive molecules, proteins and such, how do they organize, and how do these things function in order to make a biological pump that actually works. But often in doing these things, we are challenged by the limits of current technology, and we don't like that no for an answer, so we just build some new tools when we need to, and that feeds our life science research and vice versa. But once we build a hammer, we sometimes see nails elsewhere, so we also do some cancer and neuroscience work. And in doing this, for any students in the audience, don't necessarily feel bound to whatever field you're currently in. That's just your current field. I started as a chemical engineer, did a PhD in biomedical engineering, focusing on whole heart, high-speed imaging, did computational modeling in a math department, and only then became a microscopist during my second postdoc. So a little overview of the small amount of material I've packed into this talk. I'll talk about some basic physiology that we did, how we applied that to human healthcare problems in real-world contexts, what's kind of around the corner for the future of these fields on the life science side. But as I go through these, I will, in context, walk you guys through the tools we've built to make these projects feasible, whether they be imaging workflows, analysis tools, or something else beyond that. So the basic physiology bit. As I mentioned, this is the process that obsesses me. For every heartbeat, each of us has 3 billion of these brick-shaped heart muscle cells called cardiomyocytes that are shortening and relaxing. And you take 3 billion of these, coordinate them like a perfect set of dominoes. If they contract in the right sequence, you get a complicated, twisting, carol-ringing type pumping motion of the heart. If that coordination is disrupted, the heart loses rhythm. This is what we call an arrhythmia. This can be anything from, oh, it sucks to go upstairs, to I am dead in seconds. And we don't like that, so we want to understand what happens right here at the cell-cell interface when the electrical signals that prompt a muscle cell to contract are being propagated to the next cell. So this interface is going to be the subject of bulk of what I'll talk about today. It is the object of my obsession. Please come on in. So,
go ahead, so
yeah. I'm just waiting for this next slide to load because there's a massive image on it. This is the intercalated disc. This is the end of a cardiomyocyte. If this were a cardiomyocyte and I'm looking top down at it, I will see this. This was done by SCM, not by me, by people well before my time in science. But what it shows is the structure is complicated and heterogeneous. You have sort of steps and risers. On the steps, we see highly folded villi-like membrane structures, and the risers don't have the folding, they are straight. So 2 terms I will use, we're very clever when we're naming things as biologists. The foldy bits, we call them plicate regions because they have plicae, and the non-foldy bits, we call them interplicate regions because they run between the foldy bits. On this side is a light microscope image. This was done with confocal microscopy. What we've done is, and it doesn't project at the full resolution of the actual data, but what we did is took a slice, a frontal slice to a mouse heart, labeled 2 specific proteins in red is a protein called desmoglin or desmoplakin, which is part of mechanically connecting these cells together. And in white is a connexin protein that provides electrical conduits from cell to cell. And we wanted to see every single cell to cell contact in this volume of tissue. And that's what we've imaged here. And the reason for doing this is to understand the heterogeneity in the system. It's not just within each intercalated disc that there's heterogeneity. We can very clearly see, just from the size alone, but also the organization, intercalated discs from different parts of the heart look very different. Now, within the image, you see a lot of heterogeneity. Those are changes in orientation of the structure. Heart tissue has very complex arrangement of cells that leads in a section like this to these structures being found in different orientations. But to maximize our resolution, we will mostly look at them in this sort of orientation, what we call en face, where we're looking dead on at the plane of the structure so that we maximize our lateral resolution on all the heterogeneity in this structure. But when I started my postdoc, this looked like a perfect candidate for a CLEM-based attack. I thought, I'll get my structural context, I will get my molecular organization, and I can quantify heterogeneity very cleanly. And then I got into the nitty-gritty of it and realized the throughput was not nearly enough by a few orders of magnitude. I was dreaming of taking thousands of images, and I come to find out if I did conventional CLEM, that's going to take me a few decades at current technological pace. So I don't like waiting that long. So we built a new method. We cheated. Right? We gave up the thing that makes CLEM in most senses, which is direct correlation of image to image. The reason is the chemistries for electron and light microscopes are so incompatible. Aligning nanoscale neighborhoods is such an onerous challenge. Those are the things that limit throughput. So we thought, okay, what if I give that up by myself experimental throughput by putting separate sets of samples on light and electron microscopes, but use intrinsic fiducials. So structures like, for example, those electrical junctions, what's called a gap junction, is marked by the yellow stars here. Forms this nice pentalaminar electron-dense structure visible on the TEM. But that is also composed of connexin 43 protein, which I can very easily immunolabel and detect using subdiffraction light microscopy of various flavors, either confocal, where we're pushing the resolution to about 150 nanometers, or single molecule localization techniques like STORM, where we get 20 nanometer resolution. From these types of data, we can then quantify the spatial organizations of various proteins that are not visible or identifiable on the data in relation to the electron-dense structures that we can also do on the protein. So we look at things like ion channels relative to
Yeah. All right. Hopefully folks can hear me now. So this is a method we call indirect CLEM or iCLEM because the correlation is being accomplished indirectly. Here's an example application. Right? So we took this method, tried to understand the cardiomyocyte intercalated disc. Switch to Zoom. There we go. All right. So former student of mine took on this heroic challenge. She took a few thousand transmission electron micrographs, made morphometric measurements on the intercalated discs at various length scales. And the idea was, if I'm going to go off and build an intercalated disc, what are the numbers I need to realistically reproduce the structure? And so those are the numbers we quantified all the way from the overall size of the disc, which is on the order of tens of microns, down to intermembrane spacing, which is on the order of a few nanometers. Once we get all of this information, again, big problem, we like bringing friends on so that we can gang up on the problem. In this case, we need a computational modeling help from Seth Weinberg, fellow biomedical engineer. His lab designed a brand new modeling pipeline to construct finite element models of intercalated discs based on our EM-derived data. To give you a sense for how much of an advance this is for the field, the previous most sophisticated model that was incorporated into functional models pretended that this entire structure is a flat, homogeneous, three-dimensional circle. So missed a few things, or so we thought, but this is getting at the ultrastructure, but now I need to know where to position the electrogenic proteins, the ion channels, pumps, and transporters on this mesh. And this is where the light microscope is going to come in and do its thing. So when you single molecule localization microscopy, you see if the red is a sodium channel protein, the green is gap junction protein, which is already marked on our mesh. So now if we quantify where the red is relative to green, we can put it into our mesh. How do we do this? So this is an algorithm that I constructed when I was a postdoc. We're still using it for the moment. I will tell you about its impending obsolescence towards the end of the talk. But what it does is uses a density-based non-parametric clustering approach to identify clusters of molecules, fit holes, and then we do basic spatial analysis. We look for overlaps of these clusters, we look at intercluster distances, you can calculate their statistical F, G functions, replace K, things like this, but at the cluster level. And when we do that, then we get this quantified data. We do this for various ion channels and pumps of interest, specifically the ones that are active when the reaction potential, the voltage change in the muscle cell during every beat is starting. That is the point we're interested in. Pick off. So you have sodium channels bringing excitatory current. You have some potassium channels opposing that by leaking the current. And then you've got sodium potassium ATPase resetting those gradients to enable further beats to happen. And we look at all of these relative to a gap junction or an electrical junction protein connecting 43, as well as two different mechanical junction proteins, one that's part of adherence junctions and one that's part of desmosomes. So, by the way, these are just a selection of the kinds of readouts we get, which are essentially you're looking at how clusters of ion channels, in this case sodium channels, are organized as a function of the distance from the gap junction edge. Now we take those and we can put them into the model, but I have another problem. This tells me relative distributions. I don't have an absolute number on how many channels to put into a given location because the same fluorophore blinks an unknown number of times. I've got unknown amplification factors in the immunolabeling process. Long story short, this doesn't tell me exactly how many channels to put into the mesh. So we attack that from another angle yet again. Here, another collaborator from the OSU College of Pharmacy, President Watsky, performs a variety of electrical recordings from cells, ranging from a sort of very simple whole cell level recording like this, to a small membrane plaque being recorded like that, or recording the activity of individual ion channels or individual clusters of ion channels from a nanoscale using what is called scanning ion conductance microscopy guided smart patch clamp. It's a mouthful, but it's really a simple idea. It's an electrical analog to atomic force microscopy. Take a nanopipette with like a 20 nanometer bore, you pop it over the surface of the cell, you read the impedance and the feedback from the impedance is going to control the pipette. So essentially, if it reverses every time it sees high impedance, it'll map the membrane. You get a map like this. I'm not showing it in this case, but this rig also sits on top of a confocal microscope. So at the same time, we can image proteins in that cell. These two pieces of information direct us to specific sites of interest. For example, here's the intercalated disc. Now we can send the patch pipette back to this spot and say, let's record electrical current from there. And that's how we get these recordings specifically from the intercalated disc sites or the lateral surface of the cell and so on. This helps us put absolute numbers to the density of channels in some of these locations. These are on-pass images of discs on that site anyway. So we take that information, calibrate the storm data, and that then gets plugged into a computational model. Now we have a full-fledged model of the intercalated disc that underneath includes detailed ultrastructure and molecular organization. To make it computationally feasible, we then reduce this to an electrical circuit representation, which we validated does not lead to any loss of information, plug this into a tissue scale model, and predict how fast an electrical impulse will traverse that tissue. That is useful because that is a parameter we can experimentally measure. You can take heart. The convenient thing about hearts is they don't need the rest of the body to stay alive. As long as we keep them supplied with water, salt, sugars, and the right mix, they'll keep feeding. We stick it in front of a high-speed camera after we stain the heart with a voltage-sensitive dye, and then we can just optically record voltage transients in real time using a camera going about 2,000 frames a second, measure conduction velocities, and correlate back with the model and then play a fun game of removing structural features one at a time from the model to see which ones, when removed, break the predictive power of the model. This informs us of key structure-function relationships. On this slide, I won't bore you with all the details, but I'll tell you, here's the thing we found that there's no other way we could have found this other than through a combination of microscopy and computational modeling and manifold heart experiments. It turns out the upper chambers of the heart, the atria, fast impulses slower than the lower chambers. This has been known for a long time. Their cells are sized differently, and that's what mainly governs that particular feature. But when it comes to perturbations, the two chambers have different susceptibilities to have pro-arrhythmic conduction changes. And we realize that that is because underneath that cell size heterogeneity, once you account for it, it fits cell size equally across the whole tissue of the model. What we found was the upper chambers, the cell-cell contacts are actually capable of supporting faster propagation than the contacts in the lower chambers. And so even though the lower chamber conducts faster normally, when perturbed, it is more susceptible to trouble. This kind of insight seems very esoteric. It's cool. I learned some basic physiology. So what? Next step is connecting it to disease, but that's too far of a bridge to span in one shot. So we decided to construct a methodical, sort of step-by-step Rosetta Stone approach. Well, we did a failure analysis on the interpolated disk. I'm an engineer. I like simple approaches. If you give me a machine and I don't know how it works, I will start breaking it one piece at a time to see what happens. And we did that for the interpolated disk. We designed peptides that go in and selectively inhibit different adhesion structures. And we looked at structure and function. All of the work I showed you before, we repeated under these three conditions, and really also a selective control condition for each of these, where we made an inactive version of each of those peptides and tested all of that. And by doing that, here's what we realized. First off, we validated that our peptides are doing what we designed them to do. So for example, when we use this peptide that is targeting adherence junctions, we see damage at adherence junctions, but not at other sites. Quantified them here. Likewise, this peptide is damaging only gap junction adjacent regions called the Rengasayee. So essentially, we validated that we can selectively break the disk in different places. What happens when we break it? We did more strong. I genuinely thought this was a boring experiment. We were doing just the due diligence to make sure the proteins weren't doing anything interesting. But of course they were. It turns out if we disrupt adhesion, ion channels move in the membrane. And this can change physiology into pathophysiology in as little as 10 minutes. Essentially what happens in sodium channels is wherever we break an adhesion, if we break it near gap junctions, we lose a good proportion of sodium channel density near the gap junction, within 50 to 100 nanometers of that structure. If we damage mechanical junctions, we lose sodium channels from those. This is interesting because this is a signature we can potentially look for in disease if it has functional consequence. So to see if it has functional consequence, we looked at conduction velocity by whole heart optical mapping again under all these conditions. And what we realized was something quite interesting. This peptide, if I go back 2 slides, you'll see that the red peptide, NADP1, had the most egregious impact on ultrastructure of all 3 peptides. This is a structure that connects actin cytoskeletons from cell to cell. This is how the heart works together as an organ. And that is a ridiculous level of damage. And I thought that would be the most deleterious, electrically speaking. And it turned out not to be the case. Rather, the severity of functional impact turns out to be correlated to how densely were ion channels packed into the site that we destroyed or broken. And the denser the ion channel site that we target, the worse the functional impact. Now that is really the functionally relevant insight, and I'll show you how we've applied it in human health. So going to a very different scale, thinking about the whole heart again for a moment. Normally, on a given cardiac cycle, every chamber of the heart should be blood comes into the upper chambers, which gives a little, it just goes by the passive flow of gravity to the lower chambers, we give it a little extra kick, and then the lower chambers squeeze the blood through the rest of the body, which means this shouldn't be happening. You can see in this animation how many times the upper chambers are firing for each one of the beats happening in the lower chambers. This is what atrial arrhythmias look like. on the ECG, instead of one nice E wave representing atrial activity, per QRS representing ventricular activity, you get absolute chaos happening in the atria, some of which filters into the ventricles and disrupts rhythm there as well. But not at that. A few years ago, we got tissue samples from patients with AFib, put them under a transmission electron microscope, and we looked next to the gap junctions, and we realized they had nanoscale abnormality. Here is a gap junction, not reproduced terribly well in this image, but you can still see the pentalaminar structure, and you can see this yellow cleft of extracellular space is quite narrow at the edge of the gap junction. It's about 5-10 nanometers in all of our hearts, as long as we're in sinus rhythm. And the patients who show 15-20 nanometer widths in these spaces are ones with a. So we want to know, is this actually causally linked to the rhythm disturbance? Is this doing something to electrical signaling in these hearts? So to do that, another form of student of mine, Louisa, took on this crazy challenge of taking a healthy mouse and giving it AFib within an hour, mimicking what would happen to a patient on the first occasion that they experienced this arrhythmia. Somewhere, even though AFib is known to be a progressive disease, there's still a clear starting point. We don't have AFib, but some of us are going to have a moment where we suddenly start experiencing AFib. We wanted to capture and study that moment in a mouse. So Louisa looked at blood work from patients, and we identified high levels of inflammation, particularly focusing on anything related to fluid leak, because we saw a cleft space swell. We figured fluid imbalance should be contributing to that. So she identified vascular endothelial growth factor as a proximal candidate to test. And we looked at levels in AFib patients at the early stages. Turns out they have between 3 and 20 times as much VEGF in their blood as the rest of us who are healthy and good rhythm. And so the low dose here is roughly 3-fold the normal, and the high dose is roughly 20 times the normal. These are the limits found in AFib patients. When we give it to a mouse, within an hour, we see that the electrical signal propagation in their atria slows in a dose-dependent manner. And whereas if they have not seen this inflammation, they're not susceptible to atrial rhythm disturbances. This is the 60 Hz noise here. This is P wave. It's gone back to normal rhythm after we challenge it. The ones that have experienced inflammation go into AFib. So now we took the hearts from these mice, put them under our microscopes. And on the PM, we saw a worse version of exactly the same nanoscale remodeling we had identified in human patients. Here are the healthy next side. Here's what's happened in AFib patients. And you can see the same thing happening to the mouse, just to a more severe extent. That is some kind of species difference we still don't fully understand. But from our adhesion studies with the peptide, we know that if these clefts change shape, ion channels localized there could move away. So we then put these hearts under the storm system, and what we saw was here's a nice in red cloud of molecules of connexin 43, which is the gap junction. Next to it are dense clusters of sodium channels. This is a good, normal, healthy arrangement where the gap junction is flanked by dense sodium channel clusters. After an hour of experiencing inflammation, those clusters have run for their hills, they are dispersed. And when we quantify this across several hundred images, what we see is we're losing about half of the sodium channel density in these sites and about a third of the density in their mechanical junctions. So this we know is a recipe for slower, less reliable action potential propagation in this tissue. We predicted a mechanism for AFib initiation that relies on inflammation, promoting vascular leak, shoving more fluid into the heart, disrupting these nanostructures and dispersing clusters of ion channels from them, leading to defects in electrical propagation, leading to arrhythmia. And clinically, this is a known leak, although the mechanism is not understood. So right now we're trying to figure that out, so we can try and cut that leak. But we stared at it and we thought, well, this shouldn't just be selective to atria. It should also apply to ventricular arrhythmias. This is when I got a very interesting phone call from a colleague in Italy, who, without talking to me, just based on previous publications, had gone off and done a bunch of work in human patients and in mice with genetically and inherited and acquired forms of ventricular arrhythmias. And what he found, he focused here on just the green and the yellow circles. The green circles are humans without arrhythmia, and the yellow circles are people with arrhythmias. Based on our mouse measurements in the atria, we had made a prediction that intramembrane spacing greater than 40 nanometers at some of these sites would be highly deleterious. Here's our cutoff at 40 nanometers. we captured almost all of the arrhythmia patients. And we've also said normal is about 30 to 40 is the gray zone from our mouse measurements. Turns out that 30 nanometer cutoff perfectly captures the vast majority of normal patients. And the gray zone actually captures the mix of patients with and without arrhythmia. So this is very encouraging to us, and this just shows the arrhythmia curve showing that the patient with the abnormalities are much more susceptible to cardiac arrhythmias, including ones that can instantaneously kill them. So preventing these kinds of abnormalities from forming would be a very useful way to save money. That's what we want to do next. Try and figure out how do we block this from happening. Looking at this process chain, this side of it looked really hard to fight. So we picked, again, very simple idea like an engineer, it's a leak, let's plug the leak. Of course, going head to head with a cytokine is a bad idea because there's so many of them, the parallel pathways. Blocking all of them, of course, would also be a bad idea because it would ruin the patient's immunity and kill them potentially. So we needed to find a bottleneck that we could attack that is on the blood vessel wall. The blood vessel wall is an interesting structure. It's one layer of cells, the vascular endothelium. This is a barrier that determines how much fluid stays in our blood vessels and how much leaks into tissues. And this is dynamically regulated by cytokines like VEGF. So we look for bottleneck points that are downstream of the cytokine stimulating the endothelial cell, but upstream of tight junctions. These are the watertight junctions that make our blood vessels watertight. Upstream of tight junction beta. We identified two such bottlenecks, and luckily we had friends at other institutions who had developed peptides to inhibit those particular channels, which are connexin 43 hemi channels. You might recognize this from the cardiomyocyte. This protein is very ubiquitous. The 40 tissue types in the human body, only 40 of them express connexin 43. So it's everywhere, and it does various jobs. And also connexin 1 channels. We got these inhibitors, and we gave them to our mice, and we looked at what they did in structure. First, when we look at ultrastructure of these intercalated discs, near mechanical junctions, the story was kind of simple. You were just blunting the impact of the cytokine and preventing the worst damage from happening. The black curve here is intermembrane spacing between healthy membranes and an unchallenged disc. You see very low values, and you can see this in this image. These are tight spaces. If we challenge it with VEGF, we get the spread distribution, severely right shifted to much larger values. And our treatment cases are somewhere in the middle, which we can see both in the image and the summary plots. If we look near gap junctions, the story gets a little more funky. The first part remains the same. The black curve is nice left shifted, or very small values are what we see. In fact, we don't really see any measurements over 20 or so nanometers. In the disease case, we see larger values, but we see later on the emergence of a secondary mode in the distribution of this. It's a subset of sites that have been egregiously damaged. And interestingly, our therapeutic peptides did not fix the bulk of the distribution, but they prevented the emergence of this mode. So they stopped some sites from being so much more damaged than others, and this limits heterogeneity in the system, which is actually functionally good. But we also know that if we disrupt these structures, we lose ion channel from there. So the question was, have we gotten in there early enough to prevent the loss of ion channels from these structures? To do that, simultaneously took on a technical challenge here. We previously detected that change using strong molecular localization microscopy. To make it more ubiquitously doable, we wanted to see if we could do it with a confocal microscope this time. So we took confocal images of on-fast liquid discs, built a spatial analysis tool. Again, I'll go over that in a few slides time, and looked at the nearest neighbor distances of co-labeled signal components. In each of these graphs, the solid line is the observed distribution of these nearest neighbor distances, and the dashed line is what you would get if these proteins were organized completely randomly. And if we see a left shift, that implies attraction because we're seeing shorter distances than randomness would predict. A right shift would indicate repulsion between these two components. And so what we can then do is measure the deviation of observation from predicted reality under, or predicted distribution under spatial randomness, and derive a statistical metric of non-random attraction or repulsion. And we were able to show that the inflammation, which promoted arrhythmia, significantly decreased non-random attraction of sodium channels to the junctional molecules and our treatments were mitigating that thing to a significant level. So now that we know the mechanism, we then went and tested, does it actually work? Is it doing anything useful? Turns out, yes. So here are the cut points. In the untreated case, so in healthy mice, we barely see anything. We challenged 15 mice with just burst phase or caffeine and epinephrine. We only saw 5 episodes of arrhythmia across 15 months. In the case that is treated with inflammation, there's a lot of heterogeneity, but 2 more episodes from 510 across 20 mice. And with our treatments, we're bringing it back to 6 to 10 episodes roughly per group. So we know that these are peptides where we validated the mechanism do the job of preventing AFib. The problem then is how on earth we deliver a peptide drug to a human patient effectively. That is a problem because we have all kinds of defenses that when we see a foreign peptide in our blood, it gets chopped up, it's not even going to make it to the target site. So we then turn to clinically relevant small molecules that recapture the activity of these drugs. So we're exploiting off-target effects of old medication here. Naproxen is an old malaria pill that blocks the same target as ACT11. And spironolactone is an old blood pressure pill that blocks the same target as PXIL-2. Of course, you can see these are all in vivo experiments in mice. Even in mice, the small molecules are just better in vivo because we don't have this drug delivery problem. This is all published. We are now investigating. In fact, as of yesterday, we just received the results from the retrospective clinical cohort analysis, when we found that patients who were given spironolactone for other indications are actually at lower risk of AFib compared to ones on other drugs. We're now trying to start a prospective trial on spironolactone at OSU. We've submitted a grant. We're waiting to hear back about it if that happens again. So in all this, what I've shown you is clusters of ion channels are important for normal physiology. The question is why? And also the other part of the question is, if in some places clustering is necessary, are there places where clustering can be redundant? So first we started with the second question. Again, in collaboration with Professor of Biomedical, we looked at a few different disease states. First was a mutation in a regulatory protein called tau module. It's a point mutation in regulatory protein, but it turns out when a heart develops under this misregulated state, ion channels in those hearts act differently, and we saw an enrichment of NAV 1.6 sodium channel clusters. This is a different isoform from the one I talked about before, but it accumulates far too much in certain sites within 50 or so nanometers of calcium cycling proteins, along with something called sodium calcium exchanger. So essentially this blue stuff, which is over clustered in the disease, dysregulates sodium and calcium homeostasis in the heart and promotes rhythm disturbances. Turns out the same kind of thing also happens in the hearts of patients and mice who have something called Dravet syndrome. This is an inherited epileptic disorder associated with defects in sodium channels. And canonically, the thinking in the field was there's something wrong with the brain that's dysregulating the heart, and therefore these people are dying. But one thing I should mention, epilepsy is all forms of epilepsy are associated with a horrific phenomenon called SUDEP, sudden unexplained death in epilepsy, where a patient can drop dead independent of any seizure happening at that time or having happened just before. And Dravet patients have 10 times the risk of SUDEP compared to other epilepsy patients. And it turns out that's because same protein is also in the heart. It has nothing to do with the brain for the most part. If you put the defect in just the heart, you were able to recapitulate 80% of the mortality in this model, and then putting the mutation all over the body only made it 20% worse. So essentially what we realized was you have hyperclustering of these sodium channels, again, the NAV 1.6, in these critical spaces, and that is what's driving the arrhythmias in both of these cases. The good news for both of these stories is there's a drug called Veeraraghavan that is used to block this channel to treat Lou Gehrig's disease. So we figured that drug, which has been used by patients for a long time, we know it's safe. So we looked at those patients and saw, yeah, patients, ALS patients on Veeraraghavan, much better off with their AFib risk than patients on any other drug. Based on that, there's now another clinical trial underway evaluating this prospectively. This is work being done at the University of Utah with a former mentor and collaborator, Mark Monk. So what is it about these clusters that makes them so interesting? This is work from the last few months, where last fall, a barrier was kind enough to loan us a Minflux microscope. For those who are unfamiliar with it, this is a new form of single molecule localization, light microscopy, that gets a better resolution than my previous thoughts. Best case scenario, they've done this outside of cells. If you have molecules that you're looking at, they've hit 8 angstrom resolution with this microscope. But in optically dense cardiomyocytes, we were still able to get 2.5 nanometer resolution. And at this resolution, we start seeing the actual arrangements of these ion channels. You can see the red dots are a single molecule that was localized, yellow is 2, and white is 3. So essentially, we're seeing the actual clusters based on protein-protein interactions and how these things organize in terms of superclustering. So when President went and made measurements on the same scale, what we found was these red dots, the channels that are by themselves, behave very differently from the channels that are in clusters. Here's a recording from a single channel. It's closed, it opens, closes it again. You can see it keeps popping back open. This is not what a sodium channel should be doing. It should be doing this. When they're in groups, they all open together, they all close together, and they stay quiet. This is how you prevent abnormal impulses from arising in the tissue, and single channels behave differently in this respect. And this is borne out over a large number of patches. And we can show that if we reduce cluster size with a drug that inhibits forward trafficking of the protein, you can make multi-channel patches smaller and you start misbehaving them. So this gives us a sense that clustering is not only something that tunes channel behavior, but it's a tunable dial itself. Not only does it change channel behavior, it turns out it'll change how these channels respond to drugs. There are drugs that block these channels that are called use-dependent blockers, meaning they can only enter the channel when the channel is open. So the more you use the channel, the more potent the drug becomes. The single channels that were more jumpy and opening more, you can see the level of block you're getting there. The drug is way more potent than against a multi-channel cluster. So this is something that is kind of exciting and scary, because it means if your medical doctor doesn't understand what's happening to nanoscale arrangements of proteins, they might not be able to predict how drugs will act on that patient. So now we jumped it up to the whole organ level and the whole animal level. We took a mutant that you can see the wild type has nice big sodium current. This mutant has a loss of function, doesn't produce nearly as much current as it should. And this is the ensemble measurement over a range of voltages with 12 IV curve. And the mice that have this mutation, here are isochrome maps of activation. Blue is the earliest, and lighter colors are later and later activation. In the wild type, you get nice fast propagation. With the sodium channel inhibitor, you can slow it a little bit, but not by much. In the mice that are expressing this mutation, it's already slow, and it's super sensitive to any sodium channel inhibition. This in humans, this particular mutation is associated with something called Rengasayee syndrome that looks exactly like this in the patient, and they kill them in their sleep. And we were able to, reproduce the clinically relevant protorhythmic findings in this mouse. So that's looking good. They fiddle with clustering and change this phenotype. So, Daniel, this is the stuff I was telling you about where I had to do the imaging at night because the building was shaking from construction next door. We did scan imaging on myocytes. These are isolated cardiac myocytes. It's not the whole myocyte I'm imaging for the sliver in the middle of it. So I can show you how these sodium channel clusters look in an actual heart muscle cell from a mouse. It's phenomenal. We then treated it with a drug that accelerates the rate of these channels getting into the membrane. And we can make these clusters bigger and denser. So once we did that, turns out, yeah, we can actually rescue a whole bunch of this mutant phenotype just by shoving more proteins into the membrane. Not necessarily healthy protein only, right? What we're achieving is promoting protein to protein interactions in the membrane that change the biophysical behavior of those proteins, and even the unhealthy mutant protein now behaves a bit better. And since then, we've actually figured out what some of these interactions are. We're in the process of preparing that same in the molecule. But going back to trying to cure disease, okay, so here's, as I said, sodium current should open, close, and go back to 0. This is what the mutant does. And with our treatment, we can actually reverse this abnormal behavior while we're also preserving better current up here. So better synchrony means better peak current, better inactivation in electrophysiology terms. At the whole animal level, what that translates into is wild-type animal under flectinite challenge keeps its normal rhythm. This is a depot ventricular arrhythmia. This happened to me right now. I need to get rushed to the doctor within 15 minutes. So I'm going to focus. And with our drug, we prevent that arrhythmia. This is still not a great rhythm. It's a little bit off, but nothing deadly. This patient could lock themselves into the cardiologist's office at the time. That's not that. And it's not really serious arrhythmia. So you can see we have effective arrhythmia prevention simply by changing where the proteins are in the membrane relative to each other. We've been tested it in the gain of function model. We have gain of late current in this model, delta K mutation, and with complete clustering, we can remove that gain of function phenotype where abnormal peaks are forming in the mutant part, and they no longer happen in the treatment case where we've enhanced clustering. This is basically the healthcare side of the story, right? So this is what we do. We make a lot of measurements, we make a lot of careful analysis, and from that, we understand how healthy systems work, how these systems are deranged in disease, and then we figure out points of intervention to develop therapies. But in all this, the tools are essential, and with regard to analyzing stuff, we are very concerned about where type questions, because that's where microscopes excel. They're brilliant at telling us where things are. And so we need good ways of measuring them. For single molecule localization, we don't get images as much as we get coordinates in continuous space. We've got a bunch of point addresses for molecules, and we want to know how they're organized. Previously, I showed you the method that I'm using. I'll tell you the non-parametric clustering method I'm using there is VB-scan. The problem is it has quadratic time complexity. So if I double my data size, I'm quadrupling my runtime. For the data sets that I showed you, there was about 100 terabytes of raw data on a nice desktop with a fair bit of RAM. That ran for about 11.5 months. I don't like waiting that long. I mentioned that multiple times already. So last year we built a new algorithm, and I'll show that in a moment. But another thing I disliked about that approach, and the reason it took so long to compute is not this n squared. There are two hyperparameters to VB-scan. You have a search radius and a minimal density threshold that you have to set. You want to do it in an unbiased way. You can do it with bootstrapping, but that has fourth order time complexity, and that's why I've burned a year of compute time analyzing my data. And for all of that effort, I get information on a narrow range of length scales. And if I want other information on other length scales contained within my data, I have to do a whole nother class of all of these algorithms, which is tedious. I just don't like that. And it's got no information it gives me on supercluster, which I've just shown you from the miniflux data is happening, is relevant. So we started off with discrete space, the easy version of the problem. You want to know where stuff is in discrete space. I've already shown you results from this approach. I'll just quickly walk you through what we're doing there. We segment the signals of interest. And then for each pixel of one color, in this case, we're trying to localize these red dots relative to the white circles. If you're wondering what these phantoms are, they represent the cardiac muscle cell, which tends to have two nuclei. And there was a project where we were looking at mRNA trafficking out of these nuclei for these dots. So it was a phantom designed based on real data. But what we do is for each red dot, we figure out what is the shortest distance from it to the nucleus. We collect all of these nearest neighbor distances, plot them as a cumulative distribution function to get this red curve. We get this random curve. Conventionally, people do Monte Carlo simulations, which is painstaking and slow. So instead, what we did was we thought about if you randomize these signals and you effect recalculating the CDA, what would be the asymptotic limit of that ensemble average if you kept doing it? Well, eventually, these dots, by being randomized, will have evenly sampled the entire study area. So we just sample the entire study area once. Easy shortcut, we can calculate this without Monte Carlo in one step, and we validated that multiple different ways. And so we can quickly get the random distance distribution, subtract these so we get a distance difference curve. And this gets the positive deflection, so it's a fractal relationship. And we can measure the height of this peak which turns out to be the test statistic for a two-sample Kolmogorov-Smirnov test. So right off the bat, we're straight into clear hypothesis testing, and you can say, here's the non-random attraction or repulsion between your signals in your image. But what do we do with the data in contiguous space? Previously, we were doing ECM-based clustering. Instead, we've now switched to Voronoi tessellation, because this is an elegant algorithm that doesn't require any user-defined parameters. All it does is takes the study area and a number of points and tiles the study area in such a way that each tile now contains that chunk of the study area that is closest to the one point that is located inside that tile. So in other words, dots that are grouped very tightly together will get to claim very small tiles, and dots that are in empty space far away from neighbors get to claim big tiles. And this applies in any number of dimensions, which means if we quantify the size of these tiles, we get a measure of instantaneous density of these points. If they're molecules, we're looking at instantaneous density of the detected molecule. And so we can quickly transform this cluster of points into a density. And it turns out this can be discretized, as I'll show very cleanly, without losing information, rather than going straight from here, where you lose a bunch of information if you discretize it, or you lose a bunch, you burn a bunch of memory. Of course, this creates the same situation. Now we have these density curves. We have the observed density curve, that is a solid one here. To know what is true non-random clustering, we need to randomize these points again and create another density curve for a bunch of those random cases average. That's another painful, slow Monte Carlo step to create the dashed line. You can write that if we solve it in with, right? We figured if you randomly distribute points in a 3D space, you know the nearest neighbor distances will form a Poisson distribution. These tiles are formed by drawing perpendicular bisectors on those nearest neighbor lines, which means the area bounded by these should have a closed form distribution that we can predict a priori. Turns out it's a two-parameter gamma distribution. You're able to solve that. And so by solving that equation, you can simply calculate this curve without having to do any of that Monte Carlo work. Again, we validated that against Monte Carlo. It turns out we're just as precise as that. But the advantage of this over DB scan is not just that it's faster, not just that it doesn't need hyperparameters, it keeps noise from buttons, right? So we challenge this algorithm to detect these 10 clusters with increasing amounts of noise. We have, 20% as many noise points as clustered points. Of course, we can get easy detection, that's not a problem, but it does that even when there are 200% as much noise points in the data as there are data points. We went all the way up to 400%, and it does not really care because the statistical properties of the nearest neighbor length scale for distributions on a different length scale for clustered data from non-clustered data, but including discriminate clustered points, even in the presence of copious noise. And when we compare across a variety of statistical test cases, each time we're simulating 1 million points and we're looking at a bunch of different cases, 1,000 different cases for different statistical properties of clusters and non-clustered noise points. What we see is across the board, Voronoi tessellation is way more accurate than the scan, which is our previous state of the art. And the runtime is a no contest. Voronoi tessellation beats CT scan like it's not. So this is work I'm happy to show because this is the Voronoi tessellation is my former student Andy's work. The DB scan was my work, and he's made it obsolete. Nothing makes me happier. But also, what do we do with the tessellation? Okay, we have the tessellation. We've grabbed our non-random clusters. We get these masks, right? We can see where the clusters are, or we can have density masks. We can then digitize them, as I said before, without burning a ton of memory. You can do this very easily on a laptop or a desktop. Create these digital images, run it straight back into our discrete space point pattern analysis algorithm, and measure non-random spatial association between whatever signal components you've got. The other advantage of it is at the end of all this computation, you end up with this very, very intuitive plot where -1 is perfect repulsion, 0 is indifference, +1 is perfect attraction. And if you have two components, you have an A to B relationship on one axis, you have the B to A relationship reciprocal on the other axis. So you have a fully bounded coordinate space where you can actually track complex changes in spatial organization quite effectively. And in a way that would be easily accessible to people from other fields, such as say medical doctors, they could start reading it, if they see it. They wouldn't have to understand all the math behind it. So here's some examples of how Bohr tessellation does it, grabbing different types of structures. We use here standard data sets from a European group that were published in a paper comparing different analysis algorithms for the same data. The top are nuclear pore complexes, the bottom are microtubules. And we're not giving the algorithm any system. We simply submit the data, it runs the tessellation, and it tries to grab whatever it thinks are non-randomly clustered. So it does find all the pore complexes and its fibrillary structures. However, while the fibrils, the geometry is well preserved, we're kind of going a little awry with the nuclear pore complexes. We thought about it, and we realized the problem was something rather funny. There's not enough noise in the system to claim all the empty space around it. And you're seeing these projections of these long, spiky borderline tessellation cells that extend beyond our pore complex. So the answer was, let's noise it up, but thoughtfully, right? We don't want to just put random noise in. If we're trying to claim empty space, let's just put in a rectangular grid. In other words, uniformly distributed points. And what we found was, yeah, we can, if we add the right amount of noise, not only do we see the pore complex, it's going to need to see the pore, no problem. And we took those data and we measured the dimension of the pore complexes and compared them against the right ground truth. The inner diameter of these pores is estimated to be around 60 nanometers. Our peak is at 60 nanometers. The outer diameter of these pores is estimated to be around 125 to 130 nanometers. We're right about there. So, and this is with no problem-specific input from the user. And in fact, for the injection of the rectangular grid, we've actually derived analytical solutions that can bound how much noise to add, where it will start degrading, cluster detection, and so on. So we can automate that part completely as well. So this is what I want to wrap up with. Is on the physiology side, the conclusions are that companies, how they're organizing the membrane can be a make or break factor for human health. And that stuff is dynamic on time scales we did not comprehend before. In fact, I think it's an order of magnitude faster than what we think it is now. We're saying 10 minutes, but it could change in a minute. We're working on that right now. But in doing all this, really the cleanest way it seems to develop novel therapies where we understand the mechanism is to go at it as quantitatively as possible. I'll wrap up here and take any questions.
Question in the room, online, go ahead, you have a few minutes. 35 minutes, I can start tonight.
I got green, that's a lot.
Membrane down.
No. Not yet. This is where we want to do expansion storm. Or I think I can hack it instead. But that will require me kind of getting elbow deep in the microscope because I will need to visualize the membrane. We have some blinking dyes that bind to the membrane that we're testing right now. Nothing really jumping up that's brilliant yet. But the other option is use a dye like Veeraraghavan. But that needs a 385 nanometer laser that I don't have on any of my systems. Turns out none of my laser launches are compatible with that. So once I figure out how to get that laser into my sample on one of my microscopes, I'll be able to see the membrane as well. And its fluidity, actually. No questions?
This is somewhat for Mr. Dan and you, but so I was looking at the one slide where you're talking about basically fibers as well. So you can go through and basically calculate fibers all at once than previously, right?
Yeah.
So I know we've done some work, but we're basically doing work using fiber analysis. So what I'm saying, modules you have is very, very slow. So kind of my question, Dan, will kind of back me into you might not even think that this kind of stuff is very.
You can actually hire our continuous space analysis to do that, even if you're already in discrete space, because you just pull out a sparse matrix of signal positive voxels and give them as coordinates to this algorithm.
You would be, methods
So not neural networks, but we, again, for speed, I was looking for anything that wasn't iterative. So we need done drafting it. Very fast. In fact, one of the things I glossed over is, once you figure out which points are in non-random clusters, there's a second question of which points are in which clusters. Conventionally, that's a recursive search problem. Too slow. That's where we started using graph theory, and it linearizes the problem. But the entire pipeline is somewhere between linear and n-log n. But neural networks could work there, but they would slow it down. There's an ulterior motive to us going for all the mad speed. We want the algorithm to run the microscope tomorrow. We want it to be faster than image collection. This is really the reason we want for maximum speed while maintaining accuracy.
Can you do high resolution to the end analysis? And you can do that in statistical overlay that
Oh, there's no overlay. There's only overlay of distributions here.
but I guess we need to then only assume that whatever area of the device the CM results are in the same. So if you have 300 genetic things across the large area,
It's a very simple. Never use less than 1,000 images. But for that paper, we use something like 4,000 super resolved images and 7,000 images. So we thought about the heterogeneity and just swamped it with data.
then you probably have to solve the technical.
Yeah.
You need to assume that you have.
Well, these are per region. We wouldn't just go all over the part and just do 1,000 samples. No, this is for the specific region of the part and do 1,000 images from each technique. If you want to compare two different regions, that's that data set here. I mean, if you want global. So for the global thing, what we're now doing is that's a brute force approach, which kind of favor. My students are more elegant. So what we are now doing is defining or designing novel methods where we can image. I showed you that image of the entire frontal section of the mouse file. That took me about 10 days to acquire. But now we can do it in an afternoon with some tweaks to the workflow and well, not losing the So with those kinds of data, what we're doing so far, I've shown you spatial analysis being applied globally all over the world. We're now developing methods to chunk up the image either naively by just tiling it or intelligently in structure-based ways through coarse-grained segmentation and then do localized spatial analysis. So we can turn an image like that into a heat map of whatever nano or microscale properties we are looking at. And so smoothly grab the variability throughout the sample in one shot.
In material science, we have the same issue. You didn't have the same microscope across all components. And we want to actually capture this point in the whole component.
Weirdly, that's the first time I saw heterogeneity. As a fourth grader, I got to see cotton being mixed on PEM. And I had no idea there was that much interesting stuff going on in the world of cotton. And that much heterogeneity. And then you have things like additive manufacturing and conceiving where you're looking for very sort of focal defects. What might be homogeneous.
Instead of these 1,000 for the interactive lab.
Yeah.
What would be the number of images we get somewhere they have a great like, yeah.
With regular time, it's still dozens to a couple hundred to quantify that level of heterogeneity. But that's a theoretical estimate based on other people's work and some calculations I've done. Realistically, we would need to test it, which we're starting to do some of that work. So
There's like an order of magnitude more you need.
roughly, yeah, I suspect it'll still lock out about an order of magnitude. Because you are giving up that direct correlation, which means there's internal heterogeneity that is now an unknown. Yeah.
You have to have a larger area, rather than a small area like.
Oh, no. If we're willing to burn time and we can make photostable samples, like the image that I showed you didn't project well because the original image is many gigabytes. But that entire frontal section of the heart was imaged at 130 nanometer resolution. That's why it took 10 days. Now, prepping a sample that would survive 10 days of confocal imaging took a few tries.
Have we looked at using spatial correlation techniques like two-point correlation, homology, that can give you useful sort of representations of that particular model.
Yes,
So,
we haven't done much of that yet. We're starting to, but that's where we want to apply the, now that we're faster with the wide area imaging or large volume imaging, those are methods we're very keenly looking at. So that's planned work. We haven't done it yet.
so if you do the two-point correlation, first the photonic analysis, and each market block is represented by a set of 3 and 5 numbers.
Yep. And again, it turns out graph theory comes in useful there as well. We could do a monstrous dimension reduction. If we take three-dimensional data and just pretend it's a computational graph, we just brush that third dimension into a connectivity. And now your analysis is moving fast. In fact, I swiped that idea from a physicist, a biophysicist in France named Patrick Senet, who's doing this to the arrangement of alpha carbons in a complicated protein structure. They're reducing it to three-dimensional graphs and they're able to do really beautiful structural analysis.
Because with the two, we sort of limited the choose at the state side. But with the homology that we actually capture like multi-resolution.
Yep.
Give you this fingerprint connection.
Yeah, and the Voronoi tessellation output is ideal input for a lot of those algorithms. So very much on our hit list of things to play with. Any questions from Zoom?
I haven't seen it on the right.
Okay, thank you all.
Yeah, you have much you can, go for it, that's another.